Hey guys, so we're winding down chapter 17. We're going to be talking about how we get new species today, thus the title on the origin of species. So this is one of our last chapters about evolution. Because of the pandemic, I'm not, and us being so far behind, I'm not sure we're even going to get to any more. So it's either this one or the last set of notes. That's the last, so that's going to be pretty awesome for us. Let's get through this today. We're talking today about speciation. So that's a big important vocab word for today, speciation. And speciation is basically the creation of a new species. That makes sense, right? Speciation, getting a new species, speciation. And remember that that is also called macroevolution. We talked quite a bit about microevolution, which is changes in allele frequencies over time. That just basically means populations changing characteristics over time due to natural selection. Macroevolution is how we actually get from just changing species to creating new species. So it's going to be a pretty interesting set of notes. Let's do this. So the first thing we should talk about probably is, well, what is a species? And that is actually harder to define than you might think. Like we can mostly probably talk with among ourselves and decide that lions and tigers are different species. Um, but they can mate and they can create babies together, but they're still not the same species. And then dogs can mate and they can create babies with each other, and those babies can create babies, and those babies can create babies. But look how different those dogs are. But guess what? They are, this dog and this dog, the exact same species. It's crazy. So we really need to be able to define what a species is. And of course, all humans, every race, and all that stuff, we're all the same species of human. Homo sapien, we're the same species. And we'll decide, probably if you want to, you can use our definitions today and come to the same conclusion. All right, so here is the biological definition of a species. So the species definition, as far as biology is concerned, it is the ability to interbreed and produce fertile offspring, viable fertile offspring. In other words, if you're going to be the same species, you're going to have to be able to breed together. Now, every single one of these organisms can breed together. Well, scratch that. Every single one of these pairs is capable of breeding with each other, but not all of them can produce fertile offspring. Humans, when we mate together, we produce fertile offspring. Dogs, when they mate together, they produce fertile offspring. So same species, same species. But lions and tigers, when they mate together, they produce an offspring. That offspring is infertile. So they do not meet our criteria as the same species. Even though they can mate together, they are not the same species. So we'll kind of like, obviously there's some problems with that concept, some things that are icky, right? Because wolves and dogs can mate and produce fertile offspring, yet we still consider them separate species. So there's obviously some problems with this concept, and everything has an exception. Everything has an exception. Okay, so let's talk about how we actually get there. We get there through reproductive isolation, and this is a really important word for today, so definitely make sure you star it. So to get a new species, they must be reproductively isolated, and that means they can no longer successfully breed with the old species. So if these are two different species, if ever they can go back and breed with the old species, we do not consider them different species. If these guys are still capable of breeding and producing fertile offspring together, they are the same species. And once again, that really messes with like the dogs and wolves concept, right? Because it's icky. It is pretty dang icky sometimes. I think we're a little more lenient on the dogs wolves things because dogs were created artificially and have we've kind of kept their ability to breed with wolves artificially as well. I think that's why we kind of give dogs and wolves a pass. I don't know why I'm so obsessed with dogs and wolves today. I really like wolves. That's probably why. All right. So, how do we get reproductive isolation? So, first off, let's remind you of genetic drift, which is evolution being driven by random processes like a volcano suddenly killing all the brown bunnies, or a fly swatter killing all of the red flies. Just random things that cause the evolution of species. Remember, this affects small populations. So, how would a reduction in genetic variation affect the differences between populations of the same species? So, This is a way of reducing genetic variation. So how does that affect populations of the same species? It's likely if something happened to the genetic variation of a species, the differences would probably increase. They would probably become more different from each other because they'll probably start becoming separated enough to cause speciation. So let's talk about some barriers that prevent species from producing fertile offspring. This is like the how we get reproductive isolation. So there are two main types of reproductive isolation, and you're going to probably want to write a list of these. So we'll talk about a few different pre-zygotic barriers that you're going to want to write down, and we'll talk about a few different post-zygotic barriers that you're going to want to write down. Okay, so yeah, the first type of reproductive isolation barrier creator is pre-zygotic barriers. So we're going to talk about those first. The first pre-zygotic barrier that I want to talk to you about is temporal isolation. So say temporal. And I always want you to think temporal time, temporal time, temporal time. And it's more about breeding seasons, really. So this is differences in timing of the breeding cycles, and that prevents reproduction. So here's a bunch of different species of frogs, and maybe technically they could mate with each other, they could breed with each other, maybe. But notice that they each start breeding and end breeding at different times. So maybe these guys might be breeding around the same time, but these guys are definitely breeding at different times. So they're definitely not going to be able to mate with each other and produce a fertile offspring. So a lot of the times they just come into their breeding cycle later, and that's why they don't mate with each other. And that's a pre-zygotic barrier because that happens before there's ever a zygote. It prevents the zygote from ever forming. The fact that these guys are not mating at the same time of year prevents a zygote from forming. Okay. Another pre-zygotic barrier is behavioral isolation, and that's where different behaviors prevent mating from taking place. And you know this stuff. Birds, they have all kinds of showy feathers and songs and dances, and it's not just birds. Lots of species have this kind of thing going on. But these behavioral isolations really keep species from mating because they don't have the same behaviors. There's a video here of this cool dance, but of course it's not going to work. And that kind of puts me out. I want to see if we can do this cute dance, because cute dances are cute, gosh darn it. Oh, it makes me so sad. All right, well, we'll have to fix that video at another time. You just got to watch birds dance sometimes while they're mating. They're funny. But anyway, they don't ever create a zygote because they have different mating behaviors and they don't want to get together. Okay, the next one is habitat isolation. This one really messes with people sometimes because it's like seemingly such a silly difference. Habitat isolation is where they live in the same area, so they occupy different habitats within the same area, so they might not really ever encounter each other before when it's time to reproduce. Like in the same pond area, you can have two different species of garter snakes, and one will live in the water and one will live on the land, and they don't mate with each other because one lives in the water mostly and one lives on the land mostly. So they don't reproduce and they become different enough, and they're different species, and that prevents them from ever creating a zygote. So that's a pre-zygotic barrier. Then there's mechanical isolation. That's basically to do with morphological differences, like if it don't fit, it's not going to work, right? So mechanical isolation. You see this a lot with flowers, the flowers being perfectly shaped for their pollinators to come and move the sperm to the egg on the flower. So that's what's going on, is these pollinators are these flowers are perfectly shaped so that the pollinators move the sperm to the eggs in the flowers. But if they're different species and you have different species that pollinate you, you're not going to really be mating with each other because this bee is not going to go into this flower. It's the wrong type of flower for a bee. So this pollen is never going to enter the eggs of this species. So that's a pre-zygotic barrier that keeps them from mating together. And you could do this with flowers. This is talking about different flower shapes as far as like how the bees enter and stuff. It's pretty dang interesting. So mechanical isolation, if there's morphological differences that prevent them from mating with each other. They can't mate. They're shaped wrong. Okay. Then there's another pre-zygotic barrier. This is gamete isolation. So this is just basically like the sperm cannot fertilize the eggs of another species. They're not compatible. The sperm just can't get into the egg of the other species. That's gamete isolation. And of course, that's a prezygotic barrier because you can't create a zygote if the sperm can't meet the egg. This is really important in places where like you just throw your sperm out into the water and let nature take its course, like fish do, and all kinds of aquatic species just throw their sperm out. Well, luckily, a lot of that sperm and egg are incompatible, so that they don't have accidental matings going on all the time. All right, so we've talked about the prezygotic barriers. Those were all things that prevented zygotes from even forming. But it turns out that sometimes different species can mate together and they can form a zygote, but then something goes wrong after the zygote. So these would be post-zygotic barriers after the zygote. So one of the most common ones that most people know quite a bit about is reduced hybrid fertility. The hybrids are infertile. Some kind of chromosome incompatibility between the tiger and the lion. The chromosomes didn't match up together. They had different numbers. And they make the offspring infertile. So ligers, they can't mate. They have chromosome wackiness going on. Meiosis isn't going to work out, so it's not going to happen. Same with, what do you call zonkies? Yeah, zonkies and mules. They're infertile. They're perfectly healthy, maybe. Ligers aren't very healthy. Perfectly healthy individuals, but they can't mate with each other. So it's a post-zygotic barrier. So that makes a tiger and a lion a different species because their offspring is infertile. And a donkey and a horse is a different species because their offspring is infertile. Same with mules and horses, or donkeys and horses. Okay, another post-zygotic barrier is reduced hybrid viability. That just means that the hybrid zygote does not complete development. They're very frail, they're very sickly, and they die early. So it doesn't work out. Like, yay, they made a baby, but it's not a very healthy baby. It doesn't work out because of probably chromosome incompatibility or gene incompatibility of some kind. They just don't work out. And then there's hybrid breakdown. And hybrid breakdown is kind of interesting because usually the original hybrids are really robust, but then the offspring of hybrids are small or sterile. So as you go further down the line, it stops working out so well. So like you might be able to create an awesomely robust hybrid, but then its babies aren't that great. Oh yeah, it just didn't work out. Like something didn't work out further down the line. Okay, and so that happens. And if that happens, you would say, okay, they can't be the same species. They're different species. They just don't work out after a while. All right. So which of these is a prezygotic barrier? So I want you to look at all of these. Habitat, temporal, hybrid, zygote, or both A and B. And the question is, or answer is both A and B. These two are prezygotic. Male moths, oh, there's the answer. Male moths recognize females of their species by sensing chemical signals called pheromones. Is this gamete, habitat, behavioral, mechanical, or temporal? And you saw the answer was behavioral, but you know what? You could honestly make a case for mechanical as well because pheromones are chemicals, and if the pheromone it just isn't attractive to a certain species, you could call that a mechanical isolation, but also a behavioral isolation because they don't behave towards that pheromone. Honestly, a little bit of both there. Okay, so let's talk about how we get species, like the modes. How do they become isolated from each other? So we're going to talk about these three main types of speciation or modes of speciation. We'll talk a little bit about each of them, so you don't have to write them quite yet. All right, so one of the first modes of speciation is called allopatric speciation and allopatric geographic. Allopatric geographic, that's where they get separated by some kind of geographic barrier, like a river or a Grand Canyon, and they can't get to each other to mate. So a few million years later, they're not compatible anymore. They might even look very alike, but for some reason they're unable to mate with each other anymore. They just became so different over those millions of years being separated that they're unable to mate with each other anymore. So allopatric geographic is actually one of the most common ways of getting speciation. They just get separated and can't get back together. Allopatric geographic, allopatric geographic, allopatric geographic. Trying to help you there. Will help. Okay. Another type of speciation is sympatric speciation. This is where they diverge with overlapping geography. So this is allopatric, where there's a geographic barrier, but sympatric speciation, they just become their own new species right in the middle of everybody else. And so that's actually pretty rare because if you think about it, remember, you have to become a new species by not being able to breed with the old species, right? So I think how easy it would be for these guys to just kind of keep breeding after a while. Like, this is really hard to get a new species in the middle of the old species. So how exactly does that happen? It happens through polyploidy. Okay, so we'll talk about polyploidy for just a minute. So you'll probably want to say polyploidy, and that is where you get an addition of different sets of chromosomes. So you end up with more sets of chromosomes than you should. Like a diploid organism has 2 copies, and that's normal. But then you can have tetraploid organisms with 4 copies. That's not normal, but that is a way to create a new species. 6 copies of each of the chromosomes, that's a hexaploid. So yeah, it kind of is weird. And so what ends up happening, and the way they end up separated from each other, is their gametes. Because during meiosis, the gametes of a diploid organism end up haploid, but the gametes of a tetraploid organism end up diploid because they just split the DNA in half. And the gametes of a hexaploid organism end up triploid, and so on. So things get really crazy. And organisms with odd numbers of chromosomes cannot do meiosis or reproduce sexually, and so they really get all kinds of messed up, which is why bananas are really kinds of all messed up. Bananas that we eat are actually triploid. They can't mate. They can't reproduce sexually. So they're all basically clones of each other's bananas, and that's why they're having such a really hard time. Our original banana was a diploid. You can't mate them together anymore either. So it's kind of a struggle that the banana's having. They're so alike genetically that they're going extinct. So there's 2 main types of polyploidy, 2 main ways to get weird amounts of chromosomes. There is autopolyploidy, which is basically genome doubling, where you multiply the basic set of chromosomes. So you go from diploid to tetraploid, you just double it. Then there's allopolyploidy, which is the combination of distinct but similar chromosome sets. So combination of chromosome sets. Okay. Autopolyploids are derived from a single species. So when there's some meiosis that goes wrong, and you have one species with too many chromosomes and the same species with too many chromosomes, and that comes from hybridization. So check this out. This will hopefully make much more sense. So autopolyploidy is genome doubling, and that happens when there's a failure of meiosis in 2 gametes. So the egg will have too many chromosomes, and the sperm will have too many chromosomes. The sperm and egg are both diploid somehow, just because of a mistake, and so the zygote ends up being tetraploid. And that happens quite a bit. But the zygote can't go back and create haploid gametes anymore. Any gametes this one creates is diploid. So you've actually made it so this one cannot go back and mate with the originals because their gametes aren't going to be compatible anymore. Then there's allopolyploidy. They're derived, so autopolyploidy, this is when they're the same species that just double their chromosomes. But allopolyploidy is actually much more common. And what happens is you'll have just weird meiosis errors combining with not weird meiosis errors and just kind of just hybridizing weirdly. So here you have a meiosis error where you ended up having a gamete that ended up being diploid, but then you have this other gamete that was just n equals 3. You have this weird hybrid with 7 chromosomes. That does not necessarily match up, and normally this one wouldn't be viable, it wouldn't be fertile. But sometimes you can have this guy just go through meiosis, and when it does that, it actually has all kinds of issues going through meiosis, and the gamete ends up with 7 chromosomes. And then if you combine that with another normal gamete, you end up with 2 and diploid equals 10. And now you have this really weird hybrid that has an odd combination of chromosomes. And this is usually from different species hybridizing and doing really weird things with the chromosomes. If you didn't understand that, just realize it's 2 species having problems with meiosis and hybridizing because of that and doing really weird crap. And it actually is really common. We actually think that almost all of our plants are allopolyploids because plants are weird and they do all kinds of crazy stuff with their chromosomes. So plants, oh my gosh, they break so many rules. And so, I mean, it's great for them. Plants love it because polyploidy makes plants bigger, flowers bigger, fruits bigger, leaves bigger. So plants, they are very successful this way. And of course, humans have taken advantage of this. Thus are bananas and in what kinds of problems they're in. All right. Last mode of speciation is adaptive radiation. And that's just when if we have a bunch of new ecologically diverse niches, like a new habitat, you could even just say new habitat, you're going to get some new species emerging to fill in that new habitat. Like Hawaii, volcanic islands, just rock, but once plants and animals get there, they just go crazy with evolution and they adapt to the new environments, and natural selection takes over and you end up with a bunch of different new species all at once. And that's when you get new habitats. So once you get new habitats, you're going to get lots of new species. So is speciation, would you say, is it a fast or slow process? And it kind of depends. It generally takes thousands or millions of years for them to become different enough to not be able to mate with each other. But polyploidy with plants, that's super quick. So I mean, oh my gosh, all kinds of craziness, right? So what factors affect speciation rate? What factors make it go slow or fast? Well, if there's a lot of new niches, new environments, it's going to go really fast, like adaptive radiation. Bottlenecks can make things go fast. So if you lose genetic variation, they're going to become different quicker. Often when populations are isolated, so allopatric geographic isolation really helps when they're not able to get back together. So let's talk about extinction for a minute, because we talked about creating new species. Well, what factors lead to a decrease in species or an increase in extinction? And usually it's times of ecological stress, like we're going through a really great extinction right now called human impact on ecosystems, and we're killing tons of species and have really increased the extinction rate. But any other time of ecological stress, like climate change, which happens periodically on Earth, as well as being super increased right now. So yeah, lots of different things. Just environmental changes basically cause a lot of extinction. Allopatric speciation is less common than sympatric. Involves geographic isolation of some kind, is where it only happens in plants, or it requires polyploidy. Allopatric geographic isolation. All right, and then there's two models that we talk about when it comes to the creation of new species. Species can be created through a gradual change over time, or species can be created through punctuated equilibrium, which is all of a sudden quick changes that are interspersed with long periods without change. And we found that it actually happens both ways. Sometimes, depending on the environment, species might change random or gradually, or if there's a new environment or a huge shift in the environment, all of a sudden species might change quickly all of a sudden and then go through long periods without changing. So opposite ends of the continuum, both are perfectly valid forms of or ideas to explain speciation. Evolution might go into a relatively fast period as a result of all of the following except for an earthquake, a new innovation, intense competition, an eruption, or a large population. And go into a fast pace except for when there's a large population. Evolution's pretty slow when the population's pretty big. All right, guys, thank you. Hopefully this was helpful, and we will see you guys later.